Okay, so this is Chapter 12, Part 3, and we're still talking about Mendelian genetics. And so I want to start by giving you a brief recap of what we've done so far, and then talking about what we're going to be doing in this part of the chapter. So first of all, you should know by now that there are pairs of genes, right? And so diploid organisms like us, plants, Mendel's plants, we're diploids, and so genes come in pairs. We get 2 copies of each gene. And the forms of those genes are called alleles, and these are the units of heredity. And so this is what gets passed from parent to offspring in gametes, generation after generation, right? And they're passed as discrete units, alleles, that are not blended or lost in offspring. They stay as discrete units. Alleles can be dominant or recessive, right? And so dominant alleles always get expressed. Recessive alleles are only expressed in the homozygous recessive individual. In heterozygotes, we say that the dominant allele gets expressed and the recessive allele is masked or hidden, okay? Not gone, just masked, not expressed. That brings us to our first law, which we ended with at the end of Part 2, and that's the law of segregation. And so again, this is the first law that Mendel gave us. This law states that alleles for a given gene, you got 2 of them, they're going to separate from each other during meiosis, and they're going to end up in different gametes. And again, we have evidence of this from the monohybrid cross, and I also showed you this on the last slide of the last part of the chapter. Okay? Now in this part of the chapter, we're going to be talking about Mendel's second law, which is the law of independent assortment. This law states that genes that are found on different chromosomes, so that's key, they got to be found on different chromosomes, so they're not linked, they're not on the same chromosome, they're on different chromosomes. They're going to sort independently of each other during meiosis. Okay, this is going to be illustrated with what we call the dihybrid cross. So we've already seen a monohybrid cross, right? We've looked at a lot of those so far. So monohybrid is following the inheritance of just one trait. A dihybrid cross is following the inheritance of 2 traits at the same time. Remember, key here in this part of the chapter is that these 2 traits, these 2 genes, have to be on different chromosomes. Okay? Okay, so let's take a look at the law of independent assortment. And so our model organism for the first part of this chapter is going to be the guinea pig. And so we're going to be looking at 2 traits in the guinea pig. We're going to be looking at fur color and fur length. Okay, so that information is all right here. And so for color, there are 2 possible phenotypes. Guinea pigs can either be black or they can be brown, and black is dominant to brown. And so this capital B here, it represents the dominant allele, and the lowercase b represents the recessive allele. Our second trait, or second gene, which is on a separate chromosome, is the S gene, and that is going to code for fur length. We have the phenotypes of short and long. Short is going to be dominant, so we're going to use a capital letter S to represent that allele. And long will be recessive, so we will use a lowercase s to represent that allele. Okay, and so what do we have here on the right? Again, I'm showing you meiosis, and this is going to be for a guinea pig parent that is heterozygous for both genes. And so here is the genotype, big B, big S, little s. So again, for both genes, the guinea pig will be heterozygous. Now these 2 genes are on 2 separate chromosomes. Okay, this parent guinea pig will express the dominant phenotype for both genes, so black with short fur. Okay, and so here, right here is the most important part. And so this shows meiosis 1, and it shows how this can happen in 2 ways. And so this is the law of independent assortment. This says that chromosomes, okay, are going to sort independently of each other. And so therefore genes found on separate chromosomes will sort independently of each other. And so here we have the B and the S gene, right? And so if we look over here at the cell on the left first, notice how the chromosomes line up. So this homologous pair, right, containing the B genes, we have the blue chromosome containing the recessive allele on this side of the metaphase plate, and then the capital B allele, which codes for black, on this chromosome on the right side of the metaphase plate. And the same holds true for the S gene. We have the dominant over here on the right and the recessive allele over here on the left. And so look at how these chromosomes have sorted themselves. Look at the cell on the right. It's the same exact cell. It's just that the chromosomes have aligned themselves differently. So we've kind of flip-flopped the chromosomes containing the S gene. Okay, so now the dominant allele is on the left of the metaphase plate, and the chromosome containing the recessive allele is on the right-hand side of the plate. This makes a difference. This is a random event, how they line up, and there's an equal probability of the cell on the right and the cell on the left. There's an equal probability of either of these forming. But they will result in different gametes. Okay, so let's look at the cell on the left first. If this goes through meiosis 1. We end up with 2 cells that then go through meiosis 2, which give us these gametes. So this cell right here will give us these 2 gametes. The genotype of these gametes are little b, little s. This cell is going to result in these 2 gametes, which are big B, big S. These are different. Okay? This cell here on the right, again, it goes through meiosis 1, and in meiosis 2, this is what metaphase 2 looks like. It's different than what this looked like, right? Let's look at the gametes. So this cell is going to produce these 2 gametes, which are the same, which are little b, big S. And this cell right here is going to produce these 2 gametes, which are the same, big B, little s. These are 4 different gametes. So if we have a heterozygote, and it follows Mendel's rules, we will end up with 4 different gametes. Okay? Let me show you a cross. Okay, so here we go. This is an example of a dihybrid cross. And so we're going to start off with parents that are both heterozygous. Okay, and so because they're both heterozygous, we end up with this 16 boxed Punnett square. So what does this mean? So just like when we looked at the 4 boxed Punnett squares, right, the smaller ones, it's the same idea. So parental gametes down the left side and across the top, and inside these boxes are potential offspring. Okay, so how do we figure out the gametes? Well, if I look back one slide, we know it from meiosis what the gametes will be, right? So if both parents are heterozygous, we have these 4 possible gametes. If you don't want to think about meiosis all the time, you can also do this way. So let me grab my pen. Okay, so if I look at just this first parent first, and I put its gametes down the side here, this is what I would do. I would start with the B gene first, ignore the S for just a second. And if I'm looking at the B gene, I'm going to start with 1 allele, so the big B. Okay, so I'm going to say the big B allele can sort with the big S, that's possible, that's here, or the big B can sort with the little s, that's equally as likely, which is right here. Okay, I'm done with the big B allele now because I've exhausted it. That's all the possibilities. So I'll go on to my little b allele. My little b allele could end up with big S, and so that's here, or my little b allele could end up with my little s, and so that's here. And all 4 of those gametes are equally likely. So there's a quarter, a quarter, and a quarter, and a quarter possibility of each gamete forming. The same thing is going to go on the top because my other parent is exactly the same. Okay, if my other parent was not the same, it would not be the same. Okay, so what's going up here again? Big B, big S, big B, little s, little b, big S, and little b, little s. I use the lines to make sure that I got every gamete possible listed. Okay, now in the square, this is what we've already done, right? And so this first offspring that I just underlined is the result of my first sperm fertilizing my first egg, right? And so I'm going to put the B gene first and the S gene second. So this offspring is going to be big B, big S, big S. And so that's the genotype. The phenotype for that offspring, that guinea pig, is going to be black with short fur. My second box here is the result, again, of this sperm fertilizing now this egg. Okay, so we're going to have big B, big S. Now we have a little s right there. Okay, and so again, still black with short fur, but a different genotype. I'll do my third box here. So how do I get this offspring? Again, same sperm. I'm still in that first row, but now I'm in the third column over, so it's this egg. And so again, it's going to be big B, little b, big S, big S. Okay? Still black with short hair, but again, a different genotype. And I would keep going for each of these boxes. What I will end up with is this ratio down here. So let me underline that. The 9:3 to 3:1 phenotypic ratio. You should know this ratio, just like from the previous part of the chapter I said you should know the 3:1 ratio. If we're talking about a monohybrid cross, right, the 3:1 ratio is the result of 2 heterozygotes. I also told you you should be familiar with the 1:1 ratio, which is the result of 1 heterozygote and 1 homozygous recessive. Okay, here, the 9:3 to 3:1 ratio, this is a dihybrid cross in which both parents are heterozygous. That's how you're going to get this ratio. And so what does this mean? So 9/16 should show the dominant phenotype for both genes. Okay, so let me circle those. That's 1, 2, 3, 4. This one's not. 5. So any that are black short. 6, 7, 8. Nope, 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 nope. 9. Oh, nope, that's black long, sorry. Black short, that's 9. So 1, 2, 3, 4, 5, 6, 7, 8, 9. Yep, so 9/16, 9 out of 16 of the potential guinea pig offspring should show the dominant phenotype for both genes, so black short. You could then go and you can count up the next number here, which is 3 out of 16. 3 out of 16 should be black long, so dominant for the first gene, but recessive for the second. So I'll cross those out. 1, 2, and 3. The second 3 in our 9:3 to 3 ratio should be brown short, right? So recessive for color, but dominant for length. And so we've got 1 here, 2, and 3 brown shorts. And then we're left with just 1 out of the 16 that is brown long. So that guinea pig is homozygous recessive for both traits. Okay, so again, this is a ratio that you should really understand. It's a Punnett square that you should know how to build. And again, it's the result of the cross between 2 parents that are both heterozygous for both genes that we're studying. Okay, and so again, this is a dihybrid cross. I couldn't resist showing you the picture of the guinea pigs. So again, the ratio is 9:3 to 3:1, but if we're looking at our Punnett square, it's 9/16, 3/16, 3/16, and 1/16. Okay, so again, you'll see that in both lecture and lab. Now you can use a Punnett square to solve these problems, or you could use probability. And so let me show you how to do that. So let's say you don't want to construct a Punnett square. That's okay. You could also do both, construct the Punnett square and do the probability so that you have a double check. That's nice. So let's look at this problem and let's apply the rules of probability. So it says in a cross between a heterozygous black short-haired guinea pig, so I got the parents written out for us. Where's my pointer? Right here. So here is a heterozygous black short-haired guinea pig. Let's say we cross that guinea pig with a homozygous recessive pig. So that's here, my second parent. I'm asking you for the specific probability that the offspring will have this particular genotype. Big B, little s, little s. And so, let's grab my pen. So again, okay, here we go, sorry. Here it is. This is the specific genotype. And right here is our cross. So I give you the directions here. I tell you to work one trait at a time. So let's look at just the B gene. Ignore the S gene for a second. So we're really just crossing big B, little b, with little b, little b. So it's really just, that's just a monohybrid cross. And so I'm asking, what is the odds, right, that we're going to end up with a heterozygote? And so if you don't remember how to do the monohybrid cross, or you can't figure it out kind of in your head, you can go ahead and you can construct that Punnett square. And what you should find is that there's a 1/2 chance. So 1/2 of the offspring from that monohybrid cross will give us a big B, little b, the heterozygote genotype. So that's great. So we're done with that one. We can then move on to the second gene, so the S gene. So if we cross big S with little s, what are the odds of getting little s, And so again, if you construct that monohybrid cross, you'll see that the odds are 1/2. And so we take the odds of each of these individual events and we multiply them. Okay, so 1/2 times 1/2 is 1/4. Okay, and so 1/4 of the offspring are expected to be of this genotype if we have a cross between these two parents. You can do a Punnett square to also provide evidence for this. It'll be a dihybrid Punnett square. So think about how you would construct that based on what you've seen so far. You should know that this parent, we've already seen this parent, right? You can put this parent across the top. So there should be 4 types of gametes: big B, big S, big B, little s, little b, big S, and little b, little s, right? That's what we already did in the previous one. But this parent, right, this is different because we've only crossed 2 heterozygotes. This parent is homozygous recessive. So you should know that the only gametes that this parent is able to produce are homozygous recessive gametes. So only little b, little s. So there's no reason to write that 4 times. You can just write it once. So this Punnett square would only be constructed of 4 boxes. Okay, and so 1/4 of the offspring, if you do that, will have this genotype. Okay. I'm going to actually do a box like that towards the end of this lecture. So here's another question, a little bit different. So here I have 2 heterozygotes, and here is the offspring that I'm asking what the probability is. Okay, so this is, you've already seen this cross, and here is the Punnett square for that. So I'm asking you specifically, what's the probability of offspring that are big B, little s, little s. Okay, and so again, if I didn't want to construct the whole Punnett square, if I wanted to save myself some time, I could use probability. And so again, I'm going to work one trait at a time. And so I got my pen here. So I'll work with the B gene first. And so I'm going to only look here and here at the B gene. And so my question is, what are the odds of heterozygote, big B, little b? So if I cross 2 heterozygotes, I should get half of the offspring that are heterozygous. Second, what are the odds of little s, little s? So I ignore my B gene for a second. Let me erase my lines under here. And I'm going to just focus on the S gene. So I'm going to cross big S, little s with big S, little s. And only 1/4 of my offspring, do that monohybrid cross if you need to, only 1/4 of the offspring should be little s, little s. Okay, so I have a 1/2 and 1/4. I'm going to multiply those to get my probability of big B, little b, little s. And if you multiply 1/2 by 1/4, you end up with 1/8. We can double check our work here, right? We can look at our Punnett square. And so we're looking for guinea pigs that have this particular genotype of big B, little s, and so here's 1. And here is a second one. So there's only 2 out of 16, and so 2/16 is equal to 1/8. Okay, so again, you can use the Punnett square to double check your work if you choose to do probability. Using probability is nice, especially when we get beyond 2 genes. So there are trihybrid crosses. Okay, there might be a question or 2 here or there about trihybrid crosses when we get into lecture. So again, if you're trying to move kind of quickly through these problems and not spend too much time, doing the probability makes it go a little bit faster. Okay, and again, when you get beyond 2 genes to a trihybrid cross, we're looking at 3 genes. it becomes a little bit more challenging. Okay, so I just wanted to show you Mendel's work. So we're talking about guinea pigs, right? But Mendel worked with peas. And so same thing held true. So let's just quickly work through this. So Mendel always started with parental plants, right? True breeding, pure lines. And so in this case, he's looking at seed or pea shape and color. And so yellow is dominant to green and round is dominant to wrinkled. Okay, and so we're using big Y for yellow, little y for the allele for green, big R for the allele for round, and little r for the allele for wrinkled. Okay, so get your bearings on this. So in the parental generation, our two true breeding parents are both going to be homozygous for both traits. Either homozygous dominant for our yellow round or homozygous recessive for our green wrinkled. When we cross these, 100% of the offspring will be hybrid, right? Heterozygous for both. So they're going to be big Y, big R, little r. They will display the dominant phenotype for both, so yellow and round. Now, Mendel, again, very smart, ended up taking his F1 generation and allowing them to breed. And so two heterozygotes, we've already seen this one with our guinea pigs. So big Y, big R, little r, crossed with big Y, big R, little r. Here's the Punnett square. It should look really similar. So again, down the side are the gametes of one parent, and across the top are the gametes from the other parent. They are exactly the same because the two parents are the same genetically. Inside of my Punnett square are the resulting offspring. And if you count these up, you should find a 9:3 to 3:1 ratio. So again, when you see that ratio of 9:3 to 3:1, you should automatically think these parents were both heterozygous. Again, shorthand, if you don't want to do the Punnett square, there is a way to do this using probability. So let's look at this. So we have a cross between a homozygous yellow wrinkled plant. So let me grab my pen. That parent is right here, right? Homozygous yellow and wrinkled. So homozygous dominant for color and homozygous recessive for shape. We cross that plant with a plant that is heterozygous for both color and shape. So it's yellow and round, but heterozygous. And it says, what is the probability that the offspring, of having getting an offspring that is big Y, big R, little r. So heterozygous for both. So again, you start with one trait first. So we're going to focus just on the Y. So let's erase what I have written. Okay. Grab the pen. So we're going to focus on just the Y. So we're crossing big Y with big Y, little y. And the question here is, what are the odds of getting offspring that are big Y, little y? And so again, if you need to do that Punnett square for that monohybrid, you should realize that half of the offspring will be homozygous dominant and half of the offspring will be heterozygous. So half. The second question says, what are the odds of getting big R, little r? So we have to go to our second gene. So little r crossed with big R, little r. And so the same thing, half would be homozygous recessive and half are going to be heterozygous. And so here's our heterozygous and we have half. So now to get the full probability of offspring that are heterozygous for both alleles, so big Y, big R, little r, we have to multiply those two probabilities. So 1/2 times 1/2, and there is a 1/4 chance. 1 in 4 offspring should be heterozygous. Okay? Okay. So to end off this part of the chapter, I'm going to just briefly talk to you about a 2-point test cross. And so I previously introduced you to what a test cross was. Remember, this is when you have an organism that shows the dominant phenotype, but you're unsure of the organism's genotype. And so to try to figure that out, you cross the organism with a homozygous recessive organism. Okay? So a 2-point test cross is just it's a dihybrid. So you're looking at an organism that displays the dominant phenotype for both genes, but you're unsure of the genotype at those two loci. You would cross that organism with one that is homozygous recessive. Okay, so we'll go back to guinea pigs. Okay, and so we're looking at coat color and fur length, and so this is the cross, right? So let me grab my pen. So we have an organism that we know displays the dominant phenotype, but we're not sure of the genotype. So notice how I wrote this. So I put big B dash, big S dash. So what I know about this organism is that they have at least one dominant form of both genes, but I don't know the second allele. So that dash there means it could be another dominant allele or a recessive allele. It's a question mark, okay? To figure out what's there, I'm going to cross this organism with a homozygous recessive guinea pig. So little b, little s, little s. And so my question to you is, how do I get a 1-to-1 to 1 to 1 to 1, 4 ones, ratio? What must be here for these dashes? And so pause the video and go ahead and try this. And I'm going to go to my last slide here that I left blank, and I'm going to actually do this cross. And so this ratio, this 1-to-1 to 1 to 1, is the second important ratio that I need you to know for dihybrid crosses. Okay, so big B dash, big S dash. Okay, there's my guinea pig that I know the genotype, or sorry, I know the phenotype, but I don't know the genotype. I know that it's dominant for both, but I don't know what that second allele is. I'm going to cross that guinea pig with a guinea pig that is homozygous recessive for both. So here we go, little b, little s, little s. Sorry for the delay. The pen's a little slow on here. So how am I going to set up this Punnett square? So I really just have to try different genotypes here to try to figure out, what it is. So let me start with this parent, and I'm going to put this parent's gamete on the left side of the Punnett square. And so the only type of gamete that this parent is able to produce is one that is little b, little s. This organism doesn't have any other alleles, and so that's it. That's pretty easy. So on the left-hand slide, I'm only going to have one row here because I only have one potential gamete. So here it is, little b, little s. Okay, so that's it. But across the top, I got more, right? And so I'm asking, how do I get a 1, 2, 1, pen is slow, 2 more ones. So here, 1. Oh, this is painstaking. 1. Okay, so how do I get a 1-to-1 to 1 to 1 ratio? So you have to try different alleles here. Okay, so again, hopefully you pause it and you try some different stuff, but I will just tell you this is going to be a little b, and this is going to be a little s. And so let me go ahead and put the gametes across the top. So again, one potential gamete is if these two end up together. So that's going to be big B. So there's one potential gamete. So let me draw a line there. The next one is going to be big B, little s, right? So I'm going to go to the next one, big B, little s. And I'm going to put a line over the little s because big S and little s look the same. So if the capital and lowercase letter look the same, I always try to put a line just so that I make sure I tell the difference. And let me put a line there. Okay, I'm done with the big B allele. It's segregated with as much as it can. So here's my little b, big S. We practice with these, the better we get. So we're going to do lots of practice in both lecture and lab. So there's my third, and I got one more, right? that little b could end up with little s. So there's my last little line. Okay. And I actually usually do arrows, but because this pen is so slow, I'm just doing lines. So little b, little s. Okay, so all the gametes of the first parent are across the top, and the only gamete possible from my second parent is on the left. And so it's only a 4-boxed square. So again, if you're doing a dihybrid cross where you don't need 16 boxes, you don't make 16 boxes. You don't have to. So inside my Punnett square, I'm going to grab a different color, blue. I'm going to put my offspring. So my first offspring is going to be big B, little b, big S, little s. Okay, and unfortunately I forgot to put a line over on this S, so I'm going to add it really quick just to help me with record keeping. So there's my first offspring. Here's my second one, big B, little b, little s, too big, little s. Okay, and so both of these are recessive, so I'm going to quickly just put a line over it. My next one's going to be little b. Did I do this wrong? Hold on. Oh, this is a big S. Let's see. Gotta make sure I'm doing stuff right or else I screw up. I knew that was wrong. Okay. So little b, big S, a little s. I promise it'll be much faster when we do this in lecture and lab. My pen won't be so slow. My last box here is going to be little b, little s, little s. Okay, so let's see, is this a 1-to-1 to 1-to-1 ratio? There's 4 boxes, but what are they? So this first one is going to be, it's going to have black short fur. My second one is going to be black with long fur. My third one is going to be brown with short fur. And my last one will be brown with long fur. So these are 4 different phenotypes. And so yeah, this is a 1-to-1 to 1-to-1 phenotypic ratio. Okay, so you have to know that ratio. So again, you should know the 9-to-3 to 3-to-1 ratio is the result of 2 heterozygotes being crossed. And the 1-to-1 to 1-to-1 ratio is the result of a heterozygote being crossed with a homozygous recessive. Okay, so those are the 2 ratios that you really want to know for both lecture and lab. And that concludes the third part of this chapter.